I'm Sumire.
And I'm Beth.
We're going to talk about how CRISPR can be used to modify the Anopheles mosquito to try and eradicate malaria. Mosquitoes are an excellent vector for transmitting diseases because they're small, there's trillions of them, and they reproduce very rapidly. And because of this, many microorganisms have evolved to use them as their carrier. This includes the Plasmodium parasite, using the Anopheles mosquito as a carrier and causing malaria in humans. Malaria is one of the world's deadliest diseases. It's responsible for approximately 500,000 deaths a year, and also the suffering of 300 million people every year. For many years, the search for an effective prevention method has been unsuccessful. The lack of a successful vaccine is due to the complexity of the immune response to malaria and the life cycle of the Plasmodium, as it's very complex. Furthermore, the previous exposure of the Plasmodium parasite does not provide protection against future infections. Therefore, vaccines probably aren't the best way to treat malaria. As almost half the world's population is at risk of contracting malaria, there is a massive urgency for a cure. After recent breakthroughs with CRISPR and Cas9 technology, a potential prevention technique has been found, and it could eradicate malaria completely and, by extension, all vector-driven infections. CRISPR refers to a collection of viral DNA sequences within a bacterial genome that the bacteria can use to recognize reintruding viruses and cleave their genetic material. They are able to do this as their stored viral DNA can base pair and attach with the intruding viral DNA. And then the Cas9 protein snips the new strand so it's no longer able to replicate and is discarded by the cell. This mechanism can be adapted for use in permanent genome modification. By constructing a CRISPR sequence that base pairs to a specific gene of interest, Cas9 will cleave the DNA strand at that point, and this can allow short gene sequences to be inserted into that point before ligation, which can lead to the introduction of a whole new phenotype.
So, the malarial parasite Plasmodium falciparum relies on the Anopheles mosquito for its transmission to humans, where it reproduces. By creating transgenic mosquitoes carrying anti-parasite effector genes, the Plasmodium will lose their carriers to humans, and hence will lose their reproduction method, eventually leading to the extinction of this species. So how can these mosquitoes be made? Valentino Gantz and his team have already created these transgenic mosquitoes, using CRISPR-Cas9 system to insert a Plasmodium resistance vector into the mosquito. The vector was constructed with 5 important components: the Cas9 encoding gene, the guide RNA that targets the site of insertion, the homologous flanking regions for the site, a red fluorescence marker, and of course the Plasmodium resistance genes. The red fluorescence marker allows for the identification of mosquitoes with the vector successfully inserted into their genome, as the fluorescence can be observed through the mosquito's eyes. The most important part of the vector is of course the Plasmodium resistance genes, which encodes for the antibodies M1C3 and M2A10. These antibodies attack the 2 different proteins in the Plasmodium, the eukaryote protein chitinase 1 and the circumsporozoite protein. These are essential for the development of the Plasmodium sporozoites, which is the stage that the parasite is transferred to humans. The mosquitoes that have these genes for the antibodies from the inserted vectors are able to kill the Plasmodium parasites and block the transmission to humans.
So now we have a few mosquitoes that can stop the transmission to malaria, but how do we get this resistant gene to spread throughout the entire mosquito population? As the introduction of such transgenes do not confer a fitness advantage to the mosquito itself, such genes are often eliminated by natural selection, which is obviously not good because we want it to stick around. Therefore, relying on Mendelian inheritance alone is not enough to have the desired effect to spread throughout the population. Therefore, gene drive systems are necessary as it essentially forces the transgene to become dominant, despite not providing a fitness benefit to the mosquito. The gene drive system is integrated into the mosquito through the Cas9 guide RNA and the homologous flanking regions that were part of the inserted vector. These regions are all necessary for the mutagenic chain reaction, and that is the mechanism behind the gene drive. The homologous flanking regions allow the vector to bind to the genome at a specific region. As you can see in part A and B, the red sections are lining up, which are the homologous regions. And this allows insertion of the Cas9 and guide RNA segment into one of the alleles. As you can see in part C, it's being inserted into that top allele. Expression of this allele inside the cell will generate the Cas9 protein, since it's in there, which uses the guide RNA to cleave the other alleles. So here's the other allele being cleaved by the Cas9. And then the same process with the homologous flanking regions occurs to insert the Cas9 and guide RNA into the second allele. So that's now produced a homozygous individual. So now that the mutation is homozygous, and therefore any offspring from this mosquito will have at least one of the alleles with the included insert. So let's say this new homozygous mosquito mates with a wild-type mosquito. All their offspring would be heterozygous, but the mutagenic chain reaction will continue in that offspring. So the expression of its transgenic allele would lead to its wild-type allele being cleaved and mutated as well, producing another homozygous mosquito, and this continues throughout the entire population. Eventually, this would spread through the whole population. These methods have achieved transmission rates of the transgene larger than 99%, compared to a maximum of 50% from Mendelian inheritance. So linking the transgene to a gene drive construct is therefore necessary to spread this trait throughout the population, so all the mosquitoes can become transgenic and resistant to Plasmodium.
If we have this technology that has the potential to eradicate malaria, one of the world's deadliest diseases, responsible for 500,000 deaths per year, why hasn't it been used yet? Firstly, there are many ethical concerns that need to be considered, and the risks and benefits need to be weighed up. Never before have humans consciously changed the genetic makeup of a living organism on this scale. Therefore, valid concern is warranted, as once this is done, there is no going back. And genetically modifying nature on this scale can potentially lead to adverse unwanted consequences that remain unknown. This could significantly implicate biodiversity, so consideration of species interactions and the mosquito's role in the ecosystem need to be made. When considering malaria, the risk might be acceptable as the genetic modification does not majorly change the overall genome, but only a very specific part. In this proposed mechanism, the mosquito population remains intact, still filling their ecological niche, however with the reduced ability to transmit the disease. Issues associated with the release of the genetically modified population also need to be considered. Trials involving open releases have been proposed, however it would be difficult to ensure that the gene drive does not spread beyond the trial site. Potentially, the worst case scenario is that the release is ineffective and malaria isn't eradicated, which does not leave us any worse than we are now. Another issue that must be considered is that the Plasmodium could evolve resistance to the antibodies used to incite the malarial resistance, rendering this process not only completely useless, but potentially extremely dangerous. However, the use of the gene drive technology means that the sudden release of the transgenic mosquitoes would lead to large-scale genetic modification so rapidly that potentially the Plasmodium would not have time to adapt to the change. Current vector control strategies could also be used to mitigate failure in this instance. So, as previously mentioned, vaccines are unlikely to be an effective prevention strategy. CRISPR-mediated gene drive is likely to be a more successful approach in the attempt to prevent malaria. Ultimately, if the massive population modification method is proven successful, this opens up a wide range of possibilities for other diseases, starting with mosquito-borne diseases such as Zika virus, and further to other vector-borne diseases such as Lyme's disease transmitted by ticks and sleeping disease transmitted by flies. This technology has the ability to save millions of lives worldwide. So the question is it unethical to not use this technology? And are we better off using this sooner rather than